Welcome to our SCHOTT Pharmaversity, where we explore the science shaping the future of healthcare. I'm Yen, and today we are diving into the coldest extremes of pharmaceutical packaging, from minus 80 degrees to minus 180 degrees. At the center of this story is SCHOTT Topak Freeze, a syringe system engineered to perform where others fail. And joining me to unpack the science behind it is Christoph, Head of Product Management of Polymer Solutions at SCHOTT Pharma. Christoph, welcome. Great to have you here.
Thanks, Ian. It's great to be here.
So let's start with the big picture. So why is deep cold storage such a critical topic in pharma right now?
Thanks, that's a great question. The pharmaceutical landscape is shifting rapidly. We are seeing a surge in advanced therapies, mRNA vaccines, cell and gene therapies, and viral vector-based drugs. These molecules are incredibly sensitive and often require storage at minus 80 degrees or even lower to maintain their stability and efficacy.
And can you give us some real-world examples?
Absolutely. The most well-known are the mRNA COVID-19 vaccines. But now mRNA platforms are being developed for influenza, RSV, or even cancer immunotherapies. CAR-T cell therapies, which are used to treat certain leukemias and lymphomas, are also requiring cryogenic storage. And also chain therapies are using AAV or lentivirus vectors targeting conditions like spinal muscular atrophy are expanding rapidly. These all demand reliable deep cold containment.
Very interesting. And traditional packaging like glass vials or cryo bags just can't handle that?
That's partly correct, Jan. It wouldn't be fair to say that they can't handle that. But for example, for vials, the shift away from a pandemic situation where multiple doses were being sold in one vial and also used in one vial, into a prefilled syringe system, where it's a single dose, single user experience, absolutely makes sense. On the other hand, cryobags are also very difficult to handle, and that's why we developed SCHOTT Topak Freeze, a polymer-based prefilled syringe system that maintains full functionality and integrity even at minus 180 degrees Celsius.
It's really impressive. So, what makes the syringe system so resilient?
It's all about the material synergy and design. We use cycloolefin copolymers, or COC in short, for the syringe body. That polymer has a similar thermal expansion rate, closely matches our rubber components, which helps to maintain a tight seal during extreme temperature shifts, which we are also using cross-linked silicone instead of spray-on silicone, which significantly reduces the risk of particle generation, also subvisual particle generation and silicon migration.
And how did you validate that it actually works at minus 180 degrees?
That's a very good question, Jan. So for some of those tests, we could already leverage the data of minus 20 or minus 80 degrees. But some of those tests, of course, we have to run at minus 180 degrees. I will give a detailed overview in the webinar. So I will touch base on each and every single condition. But to give you some of the feedback, we ran a comprehensive test study that includes 3 thaw cycles, air transport simulation studies, headspace gas analysis using CO2 or O2, depending on the temperature, to detect microleaks if there were some. We also tested plunger movement under low pressure, mechanical stability, and drug compatibility, especially with liquid nanoparticles, which are used in many mRNA formulations.
That's a lot of testing. So, let's talk about plunger movements. That's a that's a detail most people wouldn't think about.
Right. At very low temperatures or under air transport conditions, pressure can drop or the liquid will expand, and that might cause the plunger to shift. So the place where the plunger was originally might be a different one during shipment. So we performed a very extensive DOE where we tested different fill volumes, headspace levels, and plunger designs. For example, a reduction of the headspace and using a plunger with a higher break loose and glide force significantly minimizes the movement. We even simulated worst-case scenarios using water for injection and/or placebo, depending on which was worst case.
And Christoph, can you tell me a bit more about drug safety, especially for sensitive biologics?
Certainly. We looked at extractables and leachables, of course, what we do in all of our syringe systems. Then we had a closer look on oxygen permeation, which also shifts during frozen conditions. And finally, LMP stability. Our study showed that Topac freeze performs at least as well, and in some cases even better than a standard vial, of course, not a coated vial. For example, we observed lower LMP absorption and comparable particle size and polydispersity index after long-term storage at minus 80 degrees. The long-term data by minus 100 degrees will follow.
Wow, that's really a big deal for mRNA and gene therapy developers.
Exactly. And we have also validated our syringe system maintains CCI even after 12 months of storage at minus 100 degrees with no significant changes in mechanical or optical properties, even after multiple freeze-thaw cycles. And we have a very good indication that this will be also true for minus 100 degrees Celsius storage.
Now, coming to the next one, let's talk about scalability. Is this something pharma companies can adopt quickly?
Yes, sure. Our prefilled syringe was designed especially to meet all of the requirements of high-speed fill finish packaging lines or filling lines. So Topac Freeze is available in Nest 160 with a Lilla configuration, which is the only syringe which is available in a Lilla configuration in 1 ml long within the Nest 160, which means it's integrated very easily into high-speed fill finish lines with a very low CO2 footprint and less packaging compared to Nest 100. Of course, it's also available in Nest 100, but Nest 160, we take in consideration that it would be sufficient or even better in terms of suitability on high-speed fill finish line. Also enable a very high output to a standard Lilux syringe nest. We are offering a full design documentation, extractable reports, and process capability data. And our ongoing capacity expansion, we are ready to support large-scale commercial launches.
That's great. That's great to hear. For listeners who want to dive deeper into the data and testing protocols, SCHOTT is hosting a Pharmaversity session on July 9th. So you will find the registration link in the episode description. Christoph, thank you very, very much for your insights today.
My pleasure, Jan. Thanks for having me, and I will really look forward to the Pharmaversity session and for lots of discussions.
Dear audience, that's it for this episode. If you are working on temperature sensitive therapies, this is a conversation you will want to revisit and a technology you'll want to explore. And until next time, stay curious, stay innovative, and stay cool. Bye bye.